I was looking for you, Sensei.
Hello, よろしくお願いします。
よろしくお願いいたします。
Oh, hi, Tiziana. Happy to see you. Hello to everybody. Hi, Tomoko.
Hello, Tomoko-san.
Yeah.
Wow,
Hi.
Tomoko-san, you look so lovely.
Of course, as usual. Hello, Mercedes.
Hello, Mercedes.
Happy to see you.
Hello. Happy to see you both. Thank you.
How do you feel in Holland?
I am feeling great. Yes, I have been here for 3 right now. I am very, very happy learning a lot of things.
Good. Oh, Masakurala. Hello, doctor.
Okay, so 5 o'clock and 1 minute past. I believe we can start. Lawrence, is everything okay with our technical stuff? Yes. So Lawrence is a very important guy here. Without him, we wouldn't be able to meet like this. So I want to definitely thank him, to start with thanking him. Now, I'm very glad to see you all. Hello, all. There are many people from Europe today, from Japan, other countries in Asia, and I see lots of people coming from Africa and the Middle East and so on. I'm very, very happy to see you all. This webinar, we will start, we call it Aquaphotomics Harvest webinar, because actually this is the time of harvest in Japan. And we have this year, I think this year has been unusually fruitful for people who are working in aquaphotomics. We've published a book this year, and we had for the first time edited a special issue in journal Molecules. And this webinar is organized to further show, actually to present some of the papers which were published in this special issue. Today is going to be just first webinar in our Harvest webinar series, and we will have 4 speakers. And the second webinar is going to be somewhere in end of the October, beginning of November. Today's webinar, and actually even the special issue, would not have been possible actually without the support of Tsukino Shizuku Foundation, which is our very, very long friend, and who also actually supported the papers, the works that are going to be presented today. So first I would like to just ask Professor Tsenkova, who is the main force behind all of us in this and the original creator, to just say a few words about how this all came to be. Good evening, Professor Tsenkova, please.
Hi, Elena. Thank you very much for opening and for initiating the webinar and the series of webinars that we already have in our Society of International Society of Aquaphotomics. Really, the organization that we have in the lab, thanks to Lawrence and Elena and all of you, is incredible. I think I can see more and more people attending webinars and more papers on aquaphotomics coming. And citations that we even not believe we can have, like some months we could see. The special issue has about 20 papers, and this is thanks to a great help of Tsukino Shizuku Foundation. So I would like to, I want to ask you, can I please, when we have a conference, it's so good. Yesterday I attended a conference and gave a talk in Malaysia, online conference, Zoom. And everybody was on, on the screen. So can we have everybody on the screen for a while? Let's have everybody on the screen, if possible, so that we could see each other and know who you are. And if you can do this, I'll be very, very happy.
Hello,
thank you
Liam.
very much. Thank you very much to all of you.
Hi, Professor.
Hi, hello. So It is so good to see you all. And so what we are going to, hi, hello, Valkes, and everybody, hi. So we started with Aquaphotomics International Society 5 years ago, I think. And later on, thanks to the great support of Yunosato, in Japanese it's onsen. It is spa. It is a company. It's a company that took the courage to support aquaphotomics research since 10 years ago, even more than already 12 years ago. And because of, and I could see here, Mr. Shigeoka, thank you very much for your support. And he was the supporter of the idea of having, of course, the international society, a new international society needs help, not only for research, but for the researchers to attend our conferences and to exchange and learn from each other, especially in such an interdisciplinary science. Aquaphotomics was grew up from near-infrared society, and I always connect this to Roberto and Tiziana. Always thank you for your support, and the whole Society of Near-Infrared Spectroscopy, and Carl Norris. First of all, late Carl Norris. So I would like to, we have Tomoko Miura-san with us, who is now the president of the foundation, Tsuki no Shizuku Foundation. And Tsuki no Shizuku is the name of the so famous water of Yunosato Onsen. So you could see the connection. And I would like to thank you, to thank Miura-san for her absolutely positive response, and always to young scientists and young researchers that are going to such an area like aquaphotomics that is absolutely, I think, demanding, first of all. Of course, absolutely attractive at the same time, but also very, very demanding. Please, Miura-san, could you please tell us something?
Well, thank you so much, Professor Chenkova. Hello, everyone. My name is Tomoko Matahari Miura. I'm the chairperson of the Tsuki no Shizuku Foundation. Our foundation was established in 2017, actually at the Yunosato. It was the beginning at the Yunosato, the hot springs, in Japan, to support aquaphotomics research and education opportunities. And well, we accept donations and aquaphotomics research grant requests worldwide. So for more information, please visit our website, shizuku.or.jp. And on behalf of all the supporters and members of Foundation, we would like to congratulate Professor Chenkova on publishing the very first aquaphotomics book, and also the Aquaphotomics International Society for publishing the special issue on molecules this year. It was our great honor to be able to support some of the research papers, and we want to thank many individuals and associations who made donations to make it possible. We also want to send our gratitude to presenters for taking their time to share their research with us today. Connecting the cutting edge science with the general public to share this deep wisdom of the living system revealed through the dynamic interaction of water, light, and sound is one of our missions. So after Aquaphotomics Japan Symposium 2022, we are hosting a study gathering for Japanese public on November 29th and 30th at Hot Spring Yunosato in Wakayama. So please join us, those who can. Well, thank you, Elena and Lawrence, always for putting the webinar together. And once again, congratulations to Aquaphotomics International Society, everyone. And we look forward to hearing the presentation today. Well, thank you so much.
Thank you, Mira-san. Thank you. Just to let all of you know that this year is the year for national or regional conferences. And in November 23rd and 24th, maybe Professor Hayashi could say welcome to the Japanese Aquaphotomics conference. It's unexpected. I realize that, but November 23rd, 24th, again, Yunosato is the host of their Japanese national conference. I'm sorry, somehow I didn't switch into Japanese this time. I hope this webinar will be translated in Japanese, and the Japanese translation will be presented at the event on November 29th, 30th, right?
Yes. At November 29th and 30th.
Yes.
Yes.
Okay. Again, thank you to all the presenters, and I'm really looking forward to this webinar today. Jelena, ball back to you.
Well, thank you, Professor Tenko, and thank you very much, Tomoko-san, for everything you said. Well, then, we can actually start with the webinar. And today, we are going to concentrate the webinar around the topics which are very popular in agricultural and food sector and biological systems. The next webinar is going to have more exotic topics, but these are very, very practical topics that we're going to talk about today. Every lecture will be around 15 minutes. During the lecture, please feel free to post your questions in the chat box or in the Q&A section. And the speaker, which is present here, can also, during the video, type the answers. When the video is ending, there will be like 3 minutes for each speaker to address the questions which came from the audience. Now, the first lecture today is going to be unfortunately presented by a speaker who is not available to attend. Our very, very good friend and colleague through many, many years, Professor Zoltan Kovacs from Hungary, who worked together with me and Professor Tsenkova and some other professors here at the Kobe University on germination of rice. And first we'll hear his video, and the questions are going to be answered by me if you have. So now I will ask Lawrence, can we start with the first lecture?
From Hungary. This is Zoltan Kovacs, full professor at the Hungarian University of Agriculture and Life Sciences. And in the following few minutes, I would like to talk to you briefly about one of our recent research topics, entitled Water Spectral Patterns Reveal Similarities and Differences in Rice Germination and Induced Degenerated Callus Development, which is a result of an international collaboration between colleagues from Kobe University and Hungary. Needless to say, the importance of the understanding of cell biology, not only from theoretical, but also from practical application point of view. The biochemical and morphogenetic studies study the basics of growth and development in plant cells, mainly to detect the changes in biomolecules. Therefore, most of the state-of-the-art technologies for plant cell development research rely on destructive methods and performed only in vitro on single isolated compounds. So, the use of the non-destructive spectroscopy and aquaphotomics method for in vivo investigation without any description of the system dynamics could lead to a much better understanding of the complexity of the cell development and process. So the objectives of our work were to monitor the development of cells during rice germination and seedling growth and Callus proliferation by in vivo monitoring using short-wavelength near-infrared spectroscopy and aquaphotomics, and to determine the applicability of aquaphotomics with short-wavelength near-infrared transmittance spectroscopy to detect and describe normal and abnormal cell development using rice seeds as an experimental material. For the research, we used mature seeds of rice. These seeds were dehydrated manually and surface sterilized with 70% ethanol for 5 minutes, followed by 5% commercial solution of sodium hypochlorite, and then rinsed thoroughly 3 times with sterile distilled water. 28 seeds were prepared for both rice seedling growth and Callus induction. The seeds were then placed in closed Petri dishes containing agar-based medium and kept at room temperature in a dark box throughout the experiment for the germination and rice seedling growth of the intact seeds. For the Callus formation, seeds explants after seed sterilization and planting onto in vivo tissue culture medium containing the here shown materials were used, and plant growth regulator was also used, which caused the Callus cells generation. Then these culture cells were monitored with a so-called SICA spectrophotometer, which operates in transmittance arrangement in the range between 660 nanometer to 960 nanometer. And we fixed this fiber probe in a stand, and we shine through with the light in the Petri dishes, and then we're able to monitor the seeds individually. The seeds were measured using 5 consecutive scans in the Petri dish and with 90-degree rotation in 4 different positions, resulting in 20 spectra from each of the individual seeds. And the monitoring process took place for 24 days in every other day, resulting in a total of 12 time points, and therefore the total number of spectra were 13,440. About the data analysis, we used the spectral region between 720 and 955 nanometer. I will show you some results why we decided to stick to this range. And careful pretreatments were applied after some testing. We decided to use standard normal variety and linear detrend pretreatments. The spectral data of Callus and rice seedlings samples were analyzed separately, and then as a first step after the spectral inspection, principal component analysis was used to describe the multidimensional patterns. Then partially square regression models were built to regress on the time of the growing. After that, we realized that there is a huge biological variation of the different samples. Therefore, we used a clustering analysis with hierarchical cluster analysis to form different groups based on the different natural growth rates of these cells. And then at the end, aquagrams were used to visualize how the water structure in the rice seedlings and Callus cells changes during the monitoring period. All the calculations and visualizations were performed in the R project software. So let's see our results. Here first I would like to show you through pictures how these seeds developed in the monitoring period from the 2nd day to the 4th, 5th, 6th, and so on days up to 28 days. And we can see on the left-hand side how the root and shoot got developed, how the seedlings happened, and then on the right-hand side we can see how the Callus cells got formated. Here is a summary figure about all the spectra from Callus cells and from somatic cells. And we can see there was a huge baseline fluctuation. Therefore, we needed to optimize our pre-treatment methods. And after several different steps of testing, we decided to stay with standard normal variety and linearly trend pre-treatments to remove these wavelength baseline variations. And here we can see the average spectra of the different days on the left-hand side for the Callus cells and on the right-hand side for the somatic cells. And we can see very nice features. And as you can see, the highest variation occurred between the already mentioned 730 to 870 nanometer, which can be associated to the different compounds formation during the growing and Callus formation. And if we have a look at on the second derivative spectra of the same scans spectra, then it confirms that this is the range where the majority of the variation in this short wavelength near infrared range occurred. And already here we can see some remarkable differences, especially in this region between the Callus cells and the somatic cells. In the somatic cells, we observe more variations. If we look at the principal component analysis to discover the multidimensional patterns in this spectral range and present only separately for the Callus cells, then we can conclude in these 3 dimensions that roughly we can observe 2 main clouds groups. Here with the more blue colors, we can see one big group which derives from the first part of the Callus formation period, and there is a second group which occurred after 14 days up to the end of the growing. Compared to the Callus cells, in the somatic cells we can see a much nicer pattern with the time. We can distinguish a nice continuous deviation of the different days, from blue the younger to red the more old seeds. And it means, although in Callus samples we could observe 2 big groups during these 28 days of growing, in the vegetative somatic cells we can observe a much nicer continuous tendency in the function of time. When we model with partial least square regression technique this change in the function of time. So we built the model on the growing days using the short wavelength near infrared spectral information in the range 730 to 870 nanometers. Then we could observe quite similar results to the results observed with principal components analysis, that in case of the Callus cells, there is two main groups and there is a quite high standard deviation in each days. In the somatic cells, we can see a much nicer tendency in the function of time and smaller standard deviation in each days. To reduce a little bit this high natural variation, we used HCA, hierarchical cluster analysis, to form groups. So we observe which are the cells, which are the Callus cells, and also simultaneously which are the somatic cells, which showed more similar tendency of change during this 28 days of growing, and based on, of course, based on their spectral pattern. And with this technique, we could define 3 different clusters. So we ranked our samples to 3 different groups and analyzed them separately by these 3 different clusters. And then, of course, we defined the water matrix coordinates using the previously mentioned analysis techniques. And we use these coordinates to present the water spectral patterns of these 3 main clusters of Callus cells and also the 3 main clusters of somatic cells in these aquagrams. And what we can say, in the somatic cells, although with different speed, but in each of the 3 clusters, we could recognize a very nice tendency. The younger cells showed less hydrogen bonded pattern, here starting with the red color, and then after a few days, depending on the clusters, we could observe a more strong hydrogen bonded structure, and at the end of these 28 days, a returning tendency was observed, and this turn occurred in different days in these different clusters. We could not find a similar characteristic pattern for the Callus cells. So at the end, we can conclude that the in vivo monitoring of rice seed germination and seedling growth and induced degenerated Callus formation were successfully performed using short-wavelength near-infrared spectroscopy and aquaphotomics. The quantitative models built to predict the day of the development provided better accuracy for rice seedlings growth compared to the Callus formation. We found 8 common water bands which were used to present the water matrix patterns of these Callus and somatic rice cells at the different stages of growing. And based on this, we found the water matrix of rice seedlings show 3 developmental stages. Firstly, expressing a predominantly weakly hydrogen bonded state, then a more strongly hydrogen bonded state, and finally, at the end, a weakly hydrogen bonded state occurred again. The presented findings indicate the potential of aquaphotomics for the in vivo detection of degenerated in cell development. I would like to acknowledge all the help and support, namely to the Center of Collaborative Research and Technology Development, to UNOSATO and Konono Onsen, to Kobe University NIRS Group, especially to Professor Chenkova and all the Japanese and foreign colleagues and friends who helped us in this project. And lastly, but most importantly, we are very grateful to the Chukino Shizuku Foundation for the financial support of the open access publication of our research results, because we could publish these findings in a highly reputed scientific journal called Plants. And with this support, this research result is available for the public for free of charge. So if you are interested in more details, please check Plants Journal and find our paper and read it in more details. And finally, thank you very much for your kind attention. Don't hesitate to contact us. Here you can read my email address.
Well, now I'm very sorry that Zoltan could not attend. I would really like to say thank you to him because this was a very nice at slow pace lecture, which I always like. I mean, I love when people are presenting like this, and unfortunately, I'm usually talking
This is Zoltan.
too fast. So we had one question in chat box, and I don't know actually to answer to the second question. So comment actually of Pierre model that it is interesting as it could imply that there is photonic coupling taking place. So Pierre, if you want to elaborate more on what you meant, because I don't know what you're talking about. We have some time.
So good evening, everybody. Can you hear me?
Yes.
Okay, perfect. So thanks for organizing this event. This is very special because this allows me to share what's going on in the community and to see what others are doing. So anyway, but going back to Zoltan's presentation, can you show us the PLS datasets? Maybe Lawrence can show us the slides where Zoltan presented the PLS datasets. A little bit further, because it's not numbered. Oh, there's a number, but I couldn't see the number back then. A bit further, yes. It's afterward. It's after that.
Yes.
Let's stop here. That's perfect. So if we look at the left-hand side, we see all those accumulation of red dots. And you look at if you look at the envelopes. So if you take the maxima and follow the maxima from one bar to the next, you can actually see there's an envelope, a shape. It's kind of an enclosing function. That increases and narrows down and it increases again. So it's like a pumping activity. And we have seen similar things with cells in vitro when looking with a photomultiplier. And in this case, we were using a biophotonics approach and could see how these individual seedlings, in this case were Zoltans, but in our case it was cells, started to communicate, electromagnetic communication taking place. So I'm wondering if this particular dynamics is an intrinsic property if you have a community of cells or populations that starts to feel them electromagnetically, and you should see this in the aquagrams. And you should see them all the way through it, in the sense that you should see the shifts at the very end, when Zoltan talked about the aquagrams itself. You should see that maybe you can deduct the periodicity out of that. But in fact, I was typing, so I couldn't see the aquagrams properly. It was a little bit fast.
Yes, yes, exactly. Lawrence, if you can just put the video to the end when he's showing the aquagrams. There is always, what we always see is whenever we are, so first of all, this paper worked with rice just as a model system. But with the paper, we wanted actually to show that we can recognize what is normal cell development from what is abnormal cell development. In the normal cell development, in these rice seedlings, we saw this switching between the phases of hydrogen bonded and non-hydrogen bonded water. And during this time of monitoring, during 1 month of monitoring, we saw 3 phases switching from 1 to 2. In the case
back to the first.
Yes, back to the first. In the case of Callus, which is abnormal growth, what we saw and what we think we actually saw, there was no pattern. We had just breaking at the half of this period, and this was because the cells started to die. There was no growth. So we think that this periodicity is maybe, you know, like coherent with some other, we don't know which fields are, you know, like influencing. Maybe it's even gravity or what you said about coherence with this how do you call it, photonic coupling. So maybe that's I don't know, but the point is that we always have, when something is living, you have definitely some kind of switching patterns. So this is I think that is why you see it in your cultures too. And if you even do something with plants, plant cells, whatever living biological system, you will have this again. And you can analyze, you can divide the data into these periods that you see, for example, in PLSR regression, and then do individual analysis to see what is the characteristic of each of these periods.
Yelena, there are 2 things. May I just add another question briefly?
Yes, yes.
There are 2 things that pop up in my mind. First thing is, okay, if these cells die in the Callus cells, then you should see a flash, a burst of photons emitted from these cultures. And secondly, it would be great if Zoltan would take one of these aquagrams and makes a time series out of it, so we can see the pumping activity. It can be one of these 6 aquagrams.
Yes, yes, that is wonderful. And actually, we're very interested in a time series and real-time monitoring, and we want to actually really recognize these events when they occur, as you say, this flash. And we have help from. I mean, we're hoping that we can develop more in this direction with Hilbert's transformation, that we could recognize such events. So unfortunately, we have so many data, we have so much work, and we have little help. So it takes a little bit of time, but I think we will get even with this data to do this eventually.
Can I just add something, Pierre? I have a question for you regarding this work. I think the biggest puzzle here is, why do we have different regression vectors? And you see that we have a beautiful correlation for the Callus, but not for the somatic cells. Somatic cells are those in an envelope and having a beautiful Z shape, actually, the cell development, with more than 3 phases. But we don't see this in Callus cells, and we don't really. This is something that requires more attention, and exactly what you mentioned. Probably we can use that and go deeply into the spectrum. But if you have some idea to share, great.
My first hunch, Roniana, would be that since it's a living system, there's an interacting process going on between the functional units within the seedlings and the neighboring surrounding. It's an interchange of everything, so to speak. So you have the cell division and the root formation, the rootlets that actually pour out. So it's something like what Alexander Gurevich already saw back then 100 years ago, using his UV detector system. And this seems to be a confirmation in that direction. However, this needs to be investigated properly. So we have, first of all, a living system, which means we have to take into account that there is obviously electromagnetic coupling taking place, controlling the common, let's say, dynamics of rootlet formation. So there's no single seed which is actually lagging behind, which means the other ones are cross-contaminated electromagnetically, so to speak. And this is reflected in the aquagrams. And this needs to be elaborated. At the moment, I can't tell you what exactly is going on in the aquagrams, as we need to have the time series. This would help enormously to correlate the photonic emissions with the aquagram dynamics.
Yes.
We do have it, so maybe we can all together continue working on that. Thank you.
Yeah. Thank you too.
Thank you, Pierre. So we had a few more questions, which I answered in the chat box. So please, if you're interested in knowing the answers, just read in the chat box. And there was one question that I couldn't answer. I don't know. I don't have much information about the light source. I will check that later and provide the info. Now we should actually go to another speaker. And just before that, I would like to remind people this paper is available. It is open, free of charge, and you can find it in the Plants journal. This paper is the first paper actually that was funded, whose publication was funded by Tsukino Shizuku Foundation. So thank you very much. And thank you, Zoltan. And there are a few Zoltan students here, PhD students, Balkis Saudi and Flora Vitalis, who can say that this lecture was very nice and that we are very grateful to their professor. We are sorry that he is not here now, but he will be in Japan very soon.